students next semester will be able to see that, right? You guys can't. DNA replication, right? So actually, I took out the part on recombination, which is like the very last section in the chapter. You guys don't need to worry about that. So this guy has a genetic disease, Meyer-Gorlin syndrome. So he has issues at the origins of replications, right? So causes growth abnormalities. Makes sense. Okay, so what does replication have to be able to accomplish? So it has to be really accurate. It is very accurate. But so the book throws out this stat, you don't have to memorize it, just appreciate it. If you had an error, like just 1 per million base pairs, which you would think is, not too bad, that would be a total of 6,400 mistakes. Okay, so how do you think they come up with that 6,400 number?
What, 6.4 billion? Show gene combination.
Okay, right, yeah. And that would be enough to completely mess up the cell. Okay. So the error rate is more like 1 in every 20 million. And that those errors usually get caught. So the final error rate is more like 1 in every 60 million base pairs. So it has very good fidelity. Right? It's very accurate enzyme. Also, it has to be quick. So at 1,000 nucleotides per second, this enzyme is churning out new strands of DNA. And it's just occurring at one point, especially, and we'll see in eukaryotic chromosomal replication, you have multiple replicons or portions of the chromosome that's being replicated. So after Watson and Crick came up with their model, there were basically 3 potential DNA could be replicated. One would be a conservative method. That's where the whole molecule, as a whole molecule, is being copied. It's both strands. There could be a dispersive replication method, which means that parent DNA is broken up into each of those pieces are copied, and then it's reassembled. And the third was a semi-conservative method, where the molecule is actually split into 2, and each of those 2 represents a template by which it can be copied. Because of complementary base pairing, each form that's the result, each daughter strand of DNA is going to be identical to the parent as long as there's no errors. Right, so this figure does nicely of looking at like what happens after subsequent rounds. So this is the semi-conservative model, which we know is what occurs. And you can see after the first round, you have 50% new DNA and 50% parental, and that remains after subsequent rounds. Right down here, and you look at the total, it's still 50% parental and 50% daughter. So the conservative model, though, you start to lose parental DNA rather quickly, right? You have 1 copy there in the second round of generation, that second generation that is fully parental. All right, so there was an experiment done by Messelson that used 2 forms of nitrogen. It was a heavy version of nitrogen and the normal version of nitrogen, which is N14. So they worked with E. coli, and what they did was they did initial culturing, in heavy media first, and then they transferred it to normal media, N14 media. So when I say normal, it's still radioactive so that they can pick it up, but it's closer to the normal weight of nitrogen. Right, so think about what's going to happen. The first round of replication, 50% of the DNA is going to be heavy. And as it goes through subsequent rounds of DNA replication, you're going to start to lose that heavy molecule over time. So the E. coli first in heavy, and then they transferred it to light. and then they did differential centrifugation. Right? So when you do DNA centrifugation, you can't just do it like in a form. You have to do it on a cesium chloride gradient because it's more sensitive to pick a small molecule. But they spun it down, and the heavy band, or represented in heavy DNA, sedimented out farther than the light DNA. Sure, this is the right version. Okay, so and they continued to culture it in the light medium. So they always remain, those two bands always remain, but what happened, if you can see, like the heavy is starting to transition up in the column, right? So it's becoming lighter because it's a semi-conservative molecule, right? You have parental and daughter combined together, and collectively that combination is getting lighter in terms of the sedimentary distance it's traveling. Experiment after 2 rounds of conservative replication. Conservative. Maybe 2 bands. 2 bands, right? Conservative, you've got 2 bands. You've got one heavy and one. Right. So one light and one heavy, but still 2 bands. All right, so where does replication, origin of replication, which is abbreviated OREC, and here we call it theta replication because it's circular DNA that's being replicated, and during the process of replication, the loop, the way the loop forms, it actually looks a little like the Greek symbol theta. Right, so when you're thinking of replication, consider that it can be like unidirectional, which means it can go in one direction, or it can be bidirectional. Still chromosomes, bacteria, and most species bidirectional because so you start at one origin and then you go around the circle, and then when you get point, the 2 strands fuse. Right, so this is what they mean by that theta symbol, how it looks like, and that's why they call it theta replication. So you get a replication fork, which is where the strands separate. Right, so there's a couple steps that occur here. We're going to see there's initiator proteins and DNA helicase that's involved in unwinding the DNA. And you get 2 circular DNA molecules. All right, so linear eukaryotic replication is a little different because there's a lot more DNA. There's separate chromosomes. So you end up having thousands of origins of replication. Right, so a replicated unit is what we call a replicon. And in eukaryotes, the length of that replicon is quite large, so 300,000 base pairs. There are also multiple origins of replication, right? So these represent all individual replicons, right? You can see in linear eukaryotic chromosomes, it's occurring bidirectionally. And when replicons meet, they fuse together. All right, so this table, this should be highlighted here. I'm sorry, it got screwed up. This summarizes the difference between theta and linear eukaryotic replication. Right, so in theta, it can be unidirectional or bidirectional. Linear, it's always bidirectional. Linear has many origins. Theta has 1 origin. So with linear eukaryotic replication, you have to have a template strand. This is the strand that's going to be read by DNA polymerase. and a complement strand generated. You have to have dinucleotide triphosphates. And you have to have those phosphates on there because that makes the reaction more energetically favorable. And there are enzymes and other proteins involved. And if you take cell biology here, you'll learn this in a little more advanced way. So there are some additional proteins that are kind of left out as we learn it. Okay. All right, so the most important thing to learn, memorize, and remember about DNA polymerase is that it adds new nucleotides onto the 3' end. So the 3' end is the growing end of the DNA. Okay, very important. And my recommendation to you is to learn it that way, not to learn it. It's synthesizing DNA 5' to 3' because that can get confusing in my opinion. All right, so you can see here you've got a hydroxyl group that's hanging off of the previous nucleotide that was in, or the last RNA in the primer. We haven't talked about that yet. This could be a primer right here. And the new nucleotide is losing 2 phosphates. All right, so it's the beta and the gamma phosphates that are cut off. The alpha phosphate remains, and the phosphodiester bond is forming between this hydroxyl, which is the 3' hydroxyl, and the 5' phosphate. Right, so replication can only go in one direction, 5' to 3'. That's true in both eukaryotes and prokaryotes, all living systems. So if you split the DNA molecule, right, and you start to synthesize it on one strand and also on another, you take our DNA and we open it up. Here are our base pairs. This is 5', this is 3'. Let's put a primer down here, which is the first step. All right, so the primer has polarity as well. This is 5', this is 3', this is 3', this is 5'. So where are the nucleotides going to be added on? They're going to be added here and here, because DNA polymerase can only add nucleotides to the 3' end. Enzyme that's unzipping the DNA is moving this way, though. Right, so this gradually opens up. So this strand can be replicated continuously. And because of that, it's called the leading strand. This strand here is being built in the opposite way. Right, so there's a gap here. And because of that, we call it the lagging strand. And it's made a little different segments that we call fragments. Right, so this is called a replication fork. And you can see the direction of DNA synthesis is always towards the 3' end. All right, so Okazaki, it's probably named after the Japanese guy that these occur in the lagging strand, and we call this type of replication discontinuous, short fragments. How many of you guys remember this from Bio 1? Most people do. Good. All right, discontinuous replication is a result of which property of DNA, you think? Complementary bases, anti-parallel nucleotides, because 5' to 3' on one strand, and it's opposite on the other. All right, so let's talk about a little bit. Starts at an origin of replication, and the size of that origin is about 250 base pairs. There are initiator proteins called DNA A that bind to the origin of replication and start to unwind it a little bit. So when this was initially discovered in bacteria, they didn't have the word DNA polymerase yet. They didn't know anything about helicase yet. They just started labeling these proteins DNA A and DNA B. Those are really the only two ones that you need to know at this time. So DNA A is the initiator protein, and DNA B is DNA helicase, the enzyme that is. All right, so other proteins are involved too. When you split the DNA, you break hydrogen bonds, you make the molecule unstable. It wants to go back together. Right? And so proteins come along called single-stranded binding proteins that bind to the backbone of the DNA and help stabilize it and keep it in its open orientation. Additionally, as you are you start creating strain on the molecule. So there is an enzyme called DNA gyrase. This is in bacteria, which is a type of topoisomerase. Right? Topoisomerases, if you remember from our lecture last time, are enzymes that can nick DNA and regulate the helical structure on it. So they'll nick DNA downstream where DNA helicase is going to move to and relax the molecule so that you can correctly unzip it and replicate it. All right, so there's your initiator proteins. They get bundled up where you see, and they cause a little bit of it to unwind, just enough, sorry, for DNA helicase to fit in there and more efficiently unzip it. Right, and that's called DNA B. But nowadays we call it DNA helicase. All right, so which side does it bind to? So it binds to the lagging strand template side. That's the side it sticks to as it unzips it. Then the single-stranded binding proteins come in, see here, and they keep the thing apart. Right? Without those, this would collapse. And then DNA gyrase is ahead of it, nicking it and relaxing the molecule so that it can be properly. Oh, these questions are fun. Right? Place the following components in the order in which they are first used. Course of replication. Which one's first there? Initiator protein, then helicase, single-stranded, DNA gyrase. I agree with that. I probably wouldn't ask a question like that. Tests. Good. All right. Okay, so primers, right? So there are enzymes that can synthesize nucleotides de novo. That means like without any major assistance, without a priming site. RNA polymerase is one of them. But DNA polymerase cannot do de novo synthesis. Right? It needs a primer to start from. And this is true in both eukaryotes and prokaryotes. So in eukaryotes, there is an enzyme called primase, which is a type of RNA polymerase that generates these short primers that are about 10 nucleotides long. And they're already pre-made, right? So these are existing RNA nucleotides, right? They're sitting around in the cell. And the primer comes in and it sticks onto DNA. Right? It's got an exposed 3' hydroxyl, and that can accept the first nucleotide that DNA polymerase is going to add to it. Right? So you can see your 3' hydroxyls up here, right? And the green primer coming in. And then DNA can be synthesized at the 3' end of that primer. Right? On the leading strand, just one primer. Leading strand. On the lagging strand, it's multiple primers. So if I took this here and I cut it up into 4 quadrants, right, and I labeled the polarity of the DNA, both sides, you should be able to tell me which quadrant the leading strand is, which quadrant you would find Okazaki fragments, which quadrant you would see the helicase in, all those things. Primers are synthesized where on the lagging strand? And we're dealing with lagging strand. We know we're dealing with Okazaki fragments, right? So it's at the beginning, it would be at the beginning of every fragment. Okay, so that's the initiation step, right? Once the primer's down and DNA polymerase is in, you've got initiation. Now DNA polymerase is going to travel down the molecule at 1,000 nucleotides per second, and it's going to build DNA until some sort of termination point is reached. So helicase is going to continue to unwind the DNA. Single-stranded binding proteins are going to continue to come in. DNA gyrase keeps doing its work, and on the lagging strand, DNA priming and primers. All right, so in eukaryotic, two DNA polymerases, DNA pol 3 and DNA pol 1, that you need to know. DNA pol is the enzyme that's elongating the DNA, okay? But pol 3, you have to replace those RNA DNA. So now both of these enzymes, and they also both have, they can degrade and pull off the nucleotides. So you confirm the nucleotide activity either 5 to 3 or 3 to 5 nuclease. These are exonucleases, so they're going on and they're getting the nucleotides that are on the. All right, so DNA pol 1 will come in and use its exonuclease activity to eat up the primers, and it will use its own polymerase activity to replace the primers with DNA. Right? Also, after elongation, you reach a termination point, is where a termination protein could be found. All right, so looking at pol 1 and 3, so they both have 3' to 5' exonuclease activity, but only pol 1, which is the one that's replacing the primer, has 5' to 3' exonuclease activity. So which exonuclease activity do you think is going to be used in replacing the primer? 5' to 3'. We'll see that here in a little bit. You're having trouble visualizing that. All right, this is a long slide. I'm not going to read this to you, but I'll sum it up. So what's the DNA is doing? All right, it's adding tides onto the 3' end hydroxyl of the pre-existing nucleotide. It's creating a phosphodiester bond, right? And they're involved as well. So building proteins and things like that, proteins that are guiding DNA down the molecule. Okay, so how does that exonuclease activity work? All right, so you can see here, right, there's your primer in green, right? This is on the Okazaki fragment side, the lagging strand. And that 5' to 3' exonuclease activity means it's pulling off RNAs over here at the 5' end, and it's moving towards the 3' end. Then the polymerase activity is adding it to the 3' end of the already DNA strand. Polymerase can only add to the 3' end, right? So remember that the exonuclease activity of DNA pol 1 is going to be opposite from that, right? It's going to pull nucleotides off of the 5' end. All right, so after DNA pol 1 does that, you're still going to have a nick in the DNA, right? So there's another enzyme called DNA ligase. Where did we use DNA ligase in our lab? What was that for? That do? That was involved in the cloning, right? So shuttling that thing into the plasmid, we use DNA ligase to seal that in there. So that is the final, basically the final step, creates the gap in that phosphodiester bond, seals it all together. If you didn't have that there, you would have a serious mutation in your DNA, right? RNA polymerase would not be able to travel down. All right, so a lot of tables in this chapter, spitting out a lot more information than necessary, but this is just summarizing up all of the enzymes. All right, be sure you understand initiator protein, what it's doing. You need to know all of these enzymes, but initiator protein hasn't been mentioned in a while, which is why I circled it there. Okay. Any questions about any of these enzymes? Anyone not understanding them? Stuff. Which bacterial enzyme removes the primers? Only one, right? Okay. So what another factor that increases the fidelity of DNA replication is the proofreading capability of DNA polymerase 1. Right, so over here, we said it has 5' to 3' exonuclease activity, right? Which is involved in removing the primer, and then its polymerase activity, its own polymerase activity, not DNA pol 3, comes in and replaces the primer. But to repair DNA, right, to put a correct nucleotide in where an incorrect one was inserted, it also has 5' to 3' exonuclease activity. I'm sorry, 3 to 5' exonuclease activity. So the 3 to 5' exonuclease will correct a mismatch. Okay, so that's it for prokaryotic DNA replication. Eukaryotic DNA replication has a lot of differences. We're just going to go over some of the main ones in this chapter, not get into some of the additional scaffolding proteins that are involved. So there are a bunch of different proteins that come together and bind at the origin of replication, and we're just going to term that the origin recognition complex, or ORC. Okay? So these are proteins that come in and bind and basically load the helicase onto the molecule. They're not initiator proteins like found in prokaryotes. Right, there's also this replication licensing factor, right, which basically approves DNA polymerase to come in and do its job. Checks the integrity of the system and makes sure all the scaffolding proteins are in place, and that the ORC is correctly positioned on the DNA molecule. All right, so there are a bunch of polymerases in eukaryotes. These are all of them. Right? Obviously, it can make DNA replication very complex. Now, most of them are proofreading enzymes, right? So in order to do proofreading, you have this activity, right? You have to be able to add a nucleotide in. So this type of DNA synthesis that you see over and over again on here, this translesion DNA synthesis. So these are enzymes that can pick up the changes in the shape on a DNA molecule when you have a mismatch. If you have 4 rings across, you're going to have a bulging in the DNA molecule. And all of these enzymes can detect the bulgings, or what's called an invagination, when the thing moves in, you only have 1 or 2 rings. How many rings across this be? Right, because 1 pyrimidine and 1 purine. All right, so the only ones you have to know for replication are the ones on the top there: alpha, delta, epsilon, and gamma. It's involved in the mitochondria. That's all we got to know about that. But it's alpha and delta and epsilon that do the work in the nucleus. So delta does the lagging strand synthesis, Okazaki fragments, and epsilon does the leading strand synthesis. Okay. alpha has both primase activity, right? And it has this initiation activity as well. Right? So like the DNA pull 3 in bacteria is synonymous with delta and epsilon. Synonymous, it's equivalent. All right, almost done here. Get done quick. Am I going too fast for you guys? Okay. All right, so the problem in eukaryotic replication and linear replication is on the lagging strand. So we take this out a little more. You're going to have a segment that doesn't get replicated. So this chromosomes, which are called the telomeres. And if you look over here, so this is kind of blowing up the lagging strand here. As you get to the end, you're going to be laying down more primers. And then you get to a point where you don't have any primer complementary base pairing. So you can't lay down a primer here because there's no DNA to bind it down to. So this whole segment is going to be unreplicated. Now, think about DNA as it's copied over many generations. That gap is going to get larger and larger and larger. It's going to make the ends of the chromosomes unstable. You're going to get breakage. You're going to get all sorts of problems. And eventually, it's going to interfere with genes. This is one of the theories on aging. How we age is by the breakdown of the telomeres. But that hasn't been proven. There is an enzyme that corrects all of this. That's called telomerase. Right? So telomerase has like RNA properties and protein enzymatic properties, but it looks for like these GC-rich sequences that are found in the telomeres that are, we call this like satellite regions of the chromosome. So telomerase has a protein component and an RNA component, and the RNA component will search out for the Gs, right, in these GC-rich regions, and it has a template that's already existing in the telomerase enzyme, and it will stick onto the G region, and then it will start adding nucleotides, see up here, and elongate the strand of DNA, right? So it's this enzyme that's responsible for keeping your telomeres in check. It falls off, and then you could have synthesis take place again through DNA polymerase, which is occurring here, or another primer could get added on. Either scenario is possible. Good. All right, what would be the result of an organism's telomerase if an organism's telomerase, not telomere, was mutated and non-functional? You guys think? It would shorten. Chromosomes would shorten. All right. So prepare well for Wednesday. And be sure you bring a Scantron. If you take it in the testing center, be sure you let me know by tomorrow so I can have them send a copy over. With battery. Yeah. You guys have a great night. Study hard. What do you feel like?